webinar today is presented by Joseph Cinciano from CFIA. He will present to us the work that he has been doing, and the title of his presentation is the standardized Mahalanobis. I hope I have pronounced correctly, Joseph, please correct me afterwards. Distance approach for species distribution modeling. This is something that it sounds very exciting. I don't know a lot about it, but this will be fantastic to learn from you, Joseph, more about this. I will have supporting me today the chair of the IPRRG group, Frank, will be here with us. Unfortunately, he's not going to be able to use his voice very much, but I'm sure, Frank, you'll be able to support me having all your questions and also moderating some of the questions from the participants via the chat. This webinar will be done in English. There will be no translation to any other language, and it will be, like I said, it will be available in YouTube channels. Please, if you have any questions during the presentation of Joseph, I suggest you add to the question and answer, and then I will be able to read out loud, and if it's possible, some of the participants will also have the ability to be able to speak. So for that, please raise your virtual hand. And before I forget, my name is Rose Souza Richards. I am the Vice Chair of the IPRRG, together with worked alongside with Frank and Tomasz, who sits here as well from EFSA. And so I would like to now welcome our presenter for today, Joseph, and I'll give the floor to you. Joseph, please.
Thank you, Rose. Hi, everyone. My name is Joseph Cinciano. I'm a science authority at the Canadian Food Inspection Agency, focusing on modeling and plant health. So Rose has kindly already read the title for this. So one of the purposes of giving this presentation is to get feedback from the community. So please, if you have any questions or comments on this, I'm really open to criticism at this stage because we're preparing this for publication. So the purpose here of this is to present results of a validation / ground truthing exercise, if you use that kind of language, for using the extrapolation detection approach of the Mesgerin and colleagues paper from 2014 as a species distribution model, rather than an assessment tool for species distribution models. So in case you're unfamiliar, you might ask, what is species distribution modeling? Species distribution models try to identify where a species could be or has the potential to be present. So there's two broad classes. One is process-based, and this uses experimentally derived variables and climate data to predict potential species distributions. So a very good example of a model with a good track record is climates. These models require a lot of biological research on the organism of interest to be able to use them to their full potential. On the other side, there are correlative models. So these tend to use strictly climate variable correlations with where a species is present or not present to then predict potential species distribution. So one commonly used model is called MaxEnt, and it uses a maximum entropy measure to then identify where a species might be able to persist given the climate variables. And why would we want to model species distributions? So from my perspective, I'm focused on plant pests. You can use them in other applications, but really this is going to be focused on invasive species. And modeling species distributions can help give us advanced warning. If there's a species of concern, we can model to see where it might be able to come into the country and then survey in those areas. There's also surveillance planning. By modeling where a species could potentially come into the country or where it could persist, it can help our survey biologists place traps in the right locations. And it can influence survey method design depending on the scope of the area to be surveyed. That can influence, how many traps you're going to place and the distance between traps. We can also use the results of species distribution models for risk assessment to help inform the level of risk a species might pose and to what industries. And they also help focus resources. So if we know where a pest could persist based on this climate information and this model, we can make do with fewer resources or make better use of the resources we have. In plant protection, most of us are resource constrained, and so these models can help us get the most out of what resources we do have. And all of this is to support early detection and rapid response. Now, we're going to be talking about something called Mahalanobis distance, and it requires a slightly more complicated calculation than a typical distance metric that many of us are probably familiar with. So let's think about the idea of Euclidean distance. So that's essentially calculating the hypotenuse of a triangle in either 2 or more dimensions. On the left side here, we have a plot. Let's just say the y and the x variables are 2 different climate variables. And the blue dots indicate combinations of these climate variables where this species has been observed. If we use a Euclidean distance type metric like the climate algorithm, it's going to look at how where the species observed and its climate there relative to the centroid of its climate distribution, and draw a circle like this to indicate its climate envelope. So we can see there's already some issues with using this distance metric to determine whether a species can persist in a climate, because we have all of this green area, I'll just use the colors here, where the species has never been observed. It's a new combination of climate variables. And then using this kind of a metric, we can end up with observations that really fall outside of our constraints within the model. Now, Mahalanobis distance accounts for how the climate variables covary when calculating this distance metric. And so rather than drawing this hypothetical circle to indicate the climate envelope, it draws an ellipse. And so here in this particular example, we can see that we have a tighter climate envelope that captures the observations. There are obviously still some issues where we might expect, based on the calculation, that species could exist in that climate envelope, but we still haven't observed it. But it gets rid of all of this extra range that a Euclidean or univariate based calculation would predict. So with that, let's move on to the concept of type 1 and type 2 novelty from the Mesgrin and colleagues paper. So in the Mesgrin and colleagues paper, the Mahalanobis distance is used to calculate climate suitability and type 2 novelty. But we'll start with type 1 novelty. Type 1 novelty are climate conditions outside of the observed climatic envelope for a species. So with this work, we're only working with the observed climate envelope. In this hypothetical plot here with climate variable y and climate variable x, anything in red is type 1 novelty. The species has not been observed in those climates, and it's outside of any individual climate tolerance. So the temperature could be too high, precipitation too low, and so on. Now, type 2 novelty are climate conditions within univariate bounds, but outside the multivariate bounds of the observed climate envelope. So what does that mean? If we have our climate envelope represented by this yellow ellipse, we have some sort of maximum value for x and maximum value for y and corresponding minimum values. And type 2 novelty is anything that's outside of this multivariate ellipse, but still within the minimum and maximum values of all the climate variables. So it represents a novel environment for the species, and we'll get into some discussion about what that means when we're trying to predict, or when we're trying to identify areas where a species could establish. So the approach uses a few sets of equations, and I'll just go over them briefly. On the left side is calculating a univariate distance metric to then get at type 1 novelty. So these equations ask the question, is this particular area outside of the observed climate tolerances for a given species. And so anywhere that's identified as being type 1 novel, we don't do any of the subsequent calculations on. If it's type 1 novel, the species can't persist there, according to this model. On the right side is where we calculate Mahalanobis distance. So that's represented by these d squared values, and it accounts for how the variables covary. And for this model to work, we standardize the Mahalanobis distance. What does that mean? We take some sort of a reference range. So that could be the country of origin. It could be all the observations outside of an area of interest. And we calculate the Mahalanobis distances there, and we get the maximum value in our reference range. Then, for the area we're projecting to, we divide all of those Mahalanobis distances by that maximum value in the reference range. Anything that's greater than 1 is type 2 novel, and then anything between 0 and 1 is considered climate suitable according to this. Now, Mesgren and colleagues applied this to detect extrapolation in species distribution models, trying to identify where a model is predicting a species to occur that could be type 2 novel or type 1 novel. But we thought we could apply this as a species distribution model instead. So for the overall workflow, we focused on species that have invaded North America. So we haven't yet applied it to other countries in terms of projecting these maps. And the climate data set we relied on was the CHELSA climate data. It's a very easily accessible climate data set in a raster format. We reduced the resolution, and then we selected our climate variables based on consultation with our entomology and botany teams for which variables are of interest with respect to insects and plants. On the other data input side, we used Global Biodiversity Information Facility occurrence data, GBIF data, as well as CFIA occurrence data that we had. We cleaned this data, so for some of the species, particularly plants that were a little bit more cryptic, we had an analyst go through the iNaturalist images that these observations were pulled from to identify which observations were clear identifications of the species and which ones were ambiguous or incorrect. And we also constrained the variability in the coordinates to make sure that we weren't choosing points that, say, had a 2,000 kilometer error in its latitude and longitude. From there, we split the data into training and test data. The training data was all non-North American observations, and the testing data were all observations within North America. With the training data, we then calculate our climate reference origin, and then we can calculate climate suitability. And for the climate suitability, we calculated it with each of the 5 available models that the CHELSA climate dataset uses, and then we ensembled them by taking an average. And then we used this ensemble to then test the model. So we looked at our test data and asked, are the points in the climate suitable range, the type 2 novel range, or the type 1 novel range? So for our species inclusion criteria, they had to be a regulated pest. And at first, we had no idea how many observations we would need to be certain that we were getting any reasonable test of the model. So we needed observations that would occur in at least 130 pixels of the climate raster. So approximately 10 pixels per climate variable. And each pixel is a defined area. So let's just say it's 13 by 13 kilometers. And to give you an example of how the observations are converted into pixels, say we have 13 observation points across this grid of 9 pixels. But depending on how they clustered, in this particular example, they cluster into 3 pixels. So we need even more than 130. We needed even more than 130 observations here. So that really reduced our list down. and constrained it a little bit more. But again, we didn't know how many observations we actually needed at the beginning to get any reasonable tests. For the species occurrence data, again, we incorporated observations from GBIF and where available CFIA survey data. Some of these species haven't been detected at all in Canada, so we didn't have data for those. We then eliminated observations with high coordinate uncertainty and points within bodies of water. For the bioclimatic variables, we used a combination of temperature and precipitation variables that differed a little bit between insects and plants. I'm using the short forms for the bioclimatic variables from the Chelsea dataset, but we had 7 temperature variables for insects and 7 precipitation variables for insects as well. And we had a different combination for the plants based on our expert consultation. Now, with the climate modeling, we included the 3 shared socioeconomic pathways that were available from the Chelsea data set. So this is SSP 12.6, which is low greenhouse gas emissions, net zero by 2075. We included a mid-range scenario, which had higher greenhouse gas emissions, doubling by 2100, and then a very high greenhouse gas emissions, tripling by 2075. But overall, for the results, these didn't have a huge impact on the model test. For the model ensembling, again, it was based on 5 climate models. And we took the mean of the climate suitability estimates as well as standard deviation to look at the variability. And for the most important covariate analysis from the Mesgrin et al. Paper, which identifies which climate variables could be driving the projections, we use majority voting for that aspect because it's a qualitative data set, so we can't actually average that. So for the overall model performance, we're going to go over the grouped values across all 9 species that we used. And with this table, it's broken out by the climate scenario. And then we have 3 different measures here. Multivariate accuracy is the proportion of the points that lay within the predicted climate suitable range from the model. Univariate accuracy is anything that's within the climate suitable range or type 2 novelty, because a type 2 novelty uses a univariate calculation. And false negative is where the model predicted that the species wouldn't be climate suitable there, but it has been observed there. The overall averages are pretty good for the multivariate accuracy in the mid 80% range, but the actual range of the scores varies quite a bit depending on the particular species and the circumstances. So between 44 and 100% for the SSP 12.6, and for the very high emission scenario 5-8.5, the accuracy goes down to about 29% for one of the species. If we look at the univariate accuracy, this is much higher. So that calculation on average is well into the 90% range, 98%, 97%. So we're already seeing that the multivariate climate envelope isn't performing as well as the type 2 novelty projection. And we'll get into some discussion as to why that could be. And the false negative rates, these were calculated, again, based on anything lying outside of the univariate range as well as climate suitable range. So these tend to be pretty low, around 2%, that were predicted inaccurately. Now, if we look at insects, we've got 4 species here that we included. For North American invasion points, this is in the number of pixels where it's been observed. And I'm showing the worst performing scenarios. So for spotted lanternfly, Lycorma delicatula, the worst performing scenario had a multivariate accuracy of 23%, but a univariate accuracy of 93%. And we're going to see a plot of this in a moment. The other climate scenarios did perform better. They had a multivariate accuracy of about 40-something% for Lycorma. And then for the other 3 species, the model performed pretty flawlessly. Now, if we look at the outputs that we get from this model, there's three main outputs. I first want to emphasize that these are climate suitability maps, not species distribution maps. So this is just predicting the climate suitability. It's not showing where the species has been found. Just want to add that disclaimer right there. On the far left is our plot of the novelty. So this is where we generate those projections of whether an area is climate suitable, type 1 novel or type 2 novel. Anything in red is type 1 novel. So based on the reference climate, we would predict that the species wouldn't be able to establish there. Anything that's in yellow is considered climate suitable within that multivariate context, and anything that's blue is type 2 novel. So from the Mesgerin and colleagues paper, type 2 novel would indicate potentially some kind of an acclimation happening for the species to persist there. Or it could indicate that maybe the species was constrained by biological competition in its climate of origin, and so didn't invade those climates in that region. But because it didn't have the same competition in the new environment, it could invade that. So there's a lot of different possibilities of what that means. Now, for spotted lanternfly, again, the multivariate accuracy was fairly low, but the univariate accuracy was very high. So that blue range here on the eastern side of North America, that's around the Pennsylvania area, and that's near the core range of the spotted lanternfly invasion. So we can already see that with this particular model, if we had used it before spotted lanternfly came to North America, we wouldn't have expected that to be necessarily the first place it invades. On these maps too are little points, and these are indicating where the incorrect predictions were. So we can see on the East Coast, there's quite a few incorrect predictions. So that those would be type 1 novel, but the species has been observed. And many of them cluster near the edge of a climate suitable or type 2 novel zone. This could be due to a couple of factors. It could be over-constraining the model by including too many climate variables. Although the standardized Mahalanobis distance, one of its strengths is it accounts for those climate covariations. Another explanation is that we aggregated the climate data to a much lower resolution. And so it could be that some of those averages, especially along the coast here, would include some ocean climate information, and so it would skew the projections there. But then there are some points that just lie right outside in the southern US, far away from the edges of the predicted climate suitability. Now, if we go to the middle plot, this is showing the standard deviation across the 5 climate models. And we can see there's some variability across North America, but no particularly strong patterns here. And if we go into the far right, this is the most important covariate analysis. So again, we have our climate variables represented by those short short forms. So from 1 to 11 are temperature related, 12 to 19 are precipitation. And we can see that in that type 2 novel range, it seems to be that there's a lot of precipitation influence on the prediction. So it could be that these type 2 novel areas are being driven by precipitation that's either too high or too low compared to the reference climate, rather than something like temperature. Now let's move on to plants, because plants are different than insects. We had 5 different plant species. Again, we're showing the data for the worst performing scenarios across those 3 shared socioeconomic pathways. And again, the North American invasion points represent the number of pixels, not observations. So there were many more observations than pixels. And if we look at the worst performing projection, that was for Microstegium viminium, also known as Japanese stiltgrass in its common English name. The multivariate accuracy was around 52%, but the univariate accuracy was still high, around 95%. And the other species, the model performed quite well. I will note Alopercus myoceroides only had a few invasion points in North America. And so this accuracy value, every single additional pixel could wildly change this value because of the low number of observations. So let's dive into Microstegium viminium, Japanese stiltgrass. So again, from left to right, we have our projection of climate suitability based on novelty calculations. We have our standard deviation across those climate models, and we have our most important covariate analysis. You'll note that, again, we're seeing this interesting pattern where we have a climate suitable region ringing a type 2 novel region. There are some areas near the edges where we're seeing type 1 novelty predicted, but the species is found there. So again, that could be due to issues in averaging or potential over-constraining of the model. But there is still this region in the southern US where it's quite a bit further from projected climate suitability. If we look at the variation assessed by the standard deviation, the scale bar is actually a lot lower than for spotted lanternfly, so there's a lot less variation there, but it does tend to be clustered on where there's a type 2 novelty. And if we go over to see what might be driving the type 2 novelty, it's actually quite clear for Japanese stiltgrass. It's this bio-19, this precipitation value that's driving it. So again, it could be that the precipitation is too high or too low in that area relative to what's been experienced in the reference climate, but it's not necessarily something that maybe limited the species. So we're still working out how many variables that really need to be included in this model, but dropping that precipitation value might turn that into a climate suitability, a climate suitable range. And I'd be looking for feedback on what others think about that kind of an approach. And again, to emphasize, these are climate suitability maps, not species distribution maps. So we've done this ground truthing exercise, and then we thought, how many pixels of observation, so to speak, do we actually need to get to assess whether an accuracy value reaches a certain threshold, like 80%, 90%, and so on. So we did what we're calling a minimum cell number test. So the minimum number of cells in the climate raster that are needed to get a good lock on the multivariate climate envelope to perform well on the test. And for this, in increments of 10 pixels, we randomly sampled the data from the reference climate and went from as few as 10 to, in some cases, 4,000 or 2,000 or so pixels to see if we can get that logistic curve to see where do we reach thresholds of 80% multivariate accuracy, 90%, and so on. And we assess both multivariate and univariate accuracy. So I'll just say we're looking for how many pixels do you need where the species has been observed in the reference climate to get an accurate projection of where it could invade in your projection area. So these numbers for multivariate accuracy, it was at least about 200 pixels worth of observations, but you'll note there's a lot of scripts here that indicate infinite values obtained for some species. So for some species, this logistic regression calculation failed and resulted in an infinite prediction. So that's okay. I mean, we wouldn't expect, for instance, spotted lanternfly, where the multivariate accuracy peaked around 45%. We wouldn't expect to be able to get 80% accuracy based on its reference climate from randomly sampling that reference climate. On the univariate accuracy side, we see that it can be as little as about 150 observations, but there's still a lot of variation. And there's still some species where this logistic regression failed and predicted infinite values. But overall, our initial estimate of approximately 10 pixels worth of observations per climate variable, it turns out more is better, which I think we can all agree when we're trying to model species distributions in general, more information on where the species is better. Now, there are some limitations to the testing. The data minimum for our tests was high. And we really wanted to do this for plant pathogens as well, but they generally don't have sufficient occurrence data spread across a sufficient area. So there might be a lot of observations, but they're concentrated. And with this particular model, because it's using the covariation in climate variables to constrain the projections, fewer observations leads to an underestimate of climate suitability. And for data sources, we used GBIF, but it also pulls a lot of its information from iNaturalist, and there are issues with data quality. For 2 of the plant species, we had to drop approximately 20% of the observations before doing the model because they had ambiguous or inaccurate identifications on iNaturalist. And these are ones that are considered research grade, which only requires a few users of iNaturalist to vote that that identification is correct. As well, for pathogen data, there's not a lot on GBIF yet, although it's improving. And the sources are a lot more diverse and unlinked, and don't necessarily have the same quality measures or quality assessment like coordinate uncertainty that GBIF has. So I'm hoping that with these tests of this model, I've shown you that standardized Mahalanobis distance can accurately identify where invasions can occur in North America. But these estimates are constrained by the observed climate niche. If a set of climate variables wasn't observed in the reference climate, then it's going to be predicted as some type of novelty. So it likely underestimates the potential suitability, which we see because for spotted lanternfly and Japanese stiltgrass, there were a lot of points within the type 2 novel region. And so with that, I would be happy to take any questions, any comments, any criticisms on applying this as a species distribution model. I'm looking for any sort of feedback, so happy to hear it.
Thank you so much, Joseph. This was a very, very interesting. Even, I think I learned a lot, and this is not really my field, but I think that you have done some very interesting points. I would like to invite Frank. Frank, do you have some questions for Joseph?
So let me type it in. Give me a second.
Yeah.
But yeah, I'm gonna type it. It's gonna go in a chat. Give me a sec.
Yeah.
You can see the chat, okay, Joseph?
Yes, yeah, I have it open.
For the other participants, if you want also to ask any questions, please use the question or the question and answer, or also raise your virtual hand, and I'll be able to allow you to be able to raise your question.
Okay,
Joe, can you see it? Yeah.
Yes, yeah. So Frank's question was for spotted lanternfly, any interest in trying to look at distribution of Ailanthus altissima because of the strong preference for it as a host? I hadn't considered that yet, but for some of the plant pathogens, we had been considering using this model for the hosts to try and then project climate suitability as an indirect measure, but that's actually a good, that's a really good idea. I do want to mention that with this Mahalanobis distance calculation, it doesn't need to just use climate variables. So with a lot of species distribution models, it just uses the climate variables, but we could include elevation. We could basically any data set that's spatialized. So if we imagine for a plant species that it really requires the right soil, right, for its persistence. So we could use like the soil grids data set as another variable in this calculation. So it doesn't just need to be climate, it can be like geophysical characteristics of the environment as well. But I will take this suggestion back and try running that calculation or looking at how it compares with the model output, because we could also include a host distribution as a binary variable in this Mahalanobis distance calculation. I haven't tried like a 1 and 0 variable for it, so I don't know if that would actually break the calculation. It might because it's 1 and 0, but it would be really interesting to include that host distribution. So I will take a look at that. Thank you.
So Joseph, I have a question, and it'll be a very stupid question because this is really far out from my field. But I saw that you checked Alopecuris mesoides, blackgrass, yes?
Yeah.
Big problem in different countries. In UK, for example, New Zealand, we have, well, when I was in New Zealand, as part of the person who was regulating seas, very, very hard for us to make sure that would never happen. But of course, this kind of, have you done this modeling in other countries with different numbers of presence of that particular? I'm referring to that one, but of course it could have been to any of them, and see how your model would give you the answer, and if that would be more or less compatible with what's happening on the field or not.
Yeah, so again, I presented tests that used all non-North American data to project or to North America. But I wasn't just projecting to North America, it was a global projection. So the climate suitability map was made for the globe, and then really I just cut it down to North America for the test. But it wouldn't be too hard to do the reverse and test it. Although in practice for this model, you would want to use all the observations you have, right? Yeah. So we would want to include all the all 8 pixels of the North American observations for that projection. But we could do it pretty easily. Like I have it all written up as R code, and I'm planning to package it into an R package with well-documented vignettes. But yeah, it'd be pretty straightforward to run it for other countries. It's just use North America because with Canadian Food Inspection Agency, we're concerned about Canada, and generally if things get to the US, they can eventually come to Canada. So that's why there's that focus. But again, the model was global. So it did the whole projection for the entire planet.
No, that is really interesting because I could see this being used by other governments. That would be something interesting for them to analyze, because some of the pests that you include there, they are pretty big problems, you know. Blackgrass, it is a worldwide issue. Another one would be velvet leaf. I would be interested to see how that would react to that one. That is a big problem. But yeah, it would be interesting. You done with insects and you done with plants. Would that be a possibility? Microorganisms like fungi?
Yeah, that's where we ran into, that's where we ran into issues finding enough observations. So again, the criteria were it had to be something of interest to the CFIA. it had to have enough non-North American observations, and it had to have North American observations for the test. So there were some species where there would have been enough observations to meet like our minimum cell number tests, but most of them were in North America. So a lot of pathogens were excluded, and that's just I think that's just a consequence of how and pathogens includes microorganisms and fungi. So I think it's just a consequence of how people have typically reported them in the past. It's and you can't see all of them, right? You don't go out into a forest and say, oh, that's a bacteria there that's
No, you need to
pathogenic.
test and you
So,
need to identify and make sure. But that would be interesting for some of the big problems that we have. You're right. I cannot defend the criteria that it was used because the CFIA is behind. But when we think about the devastation that Xylella fastidiosa is causing in Italy, I wonder
oh
if
yeah.
some things like this would be interesting. And there is some other ones, for example, Fusarium cincinato, big problem in Pinus, in Pinus trees everywhere. So I just wonder if in the future, perhaps the criteria could be changed and perhaps you could look into some other types of regulated pests.
And I'd be open to collaborating with others in different countries because the model runs pretty easily. And I do want to say for pathogens, we did encounter some of these issues where the observation we couldn't get point observation. It would just be like county or a region. And I think that's because there's a lot more data sensitivity, it seems, around pathogens. And so through some of these collaborations, we could probably apply it to some of the pathogens we overlooked because those point data are there. It's just there's privacy concerns about who has access. So
okay, so that's good to know. So there is the data. What there isn't is the ownership of the data is not clear.
yeah, and I think that's more of a problem for pathogens than species we can see with our eyes, right?
Okay. There is a question on here for you to say that did somebody experience Mahalanobis approach in Africa?
So again, the projections were global. The test was done strictly for North America, but it would be really easy to apply it to the African continent as well, or a particular country. I'm working on streamlining the code so it doesn't it doesn't always do a global projection. It runs in like 15 seconds on a computer, but if you're doing anything high throughput, that can extend to hours. So I want to make it so if you just have a country of interest, you can just project there and then ignore the rest.
So is this tool available to everybody? How is the deployment of this tool?
So I'll be packaging it into an R package. That's going to be the plan. So I don't know if that will come out with the publication. There will at least be some R scripts submitted with the manuscript, but ideally I want it to be an R package. So then you can just bring it in with, if you want to use it with other species distribution models, then you have that. You can bring them together and integrate them.
That's good. Frank, any further questions for you on the chat? Can you see, Joseph, with my
yeah, I can see. So,
yeah.
So Frank's next question is with Microstagium viminium, so that's Japanese stiltgrass. Not sure about Bio-19. Mean monthly precipitation in the coldest quarter. Since seeds germinate late winter, early spring, cold quarter precipitation might be at least moderately important. Is there any practical surrogate or substitute variable? That I'm not sure about. Again, we limited ourselves to the Chelsea data set, so there may be better climate variables to use than what's in the Chelsea data set. And I will also admit that we used the climate variables irrespective of the biology of the particular species. So it was using the climate variable set, the same ones for all the insects, same ones for all the plants. So they were on kind of an equal footing for this test. But I would hope that if you have a particular species of interest, you look at its biology and you identify which climate variables are important for that species and which ones as well aren't important. Because if you include ones that aren't important, you're going to get a greater constriction on the climate envelope. But you still can include, at least right now, it's my opinion that you can still include more climate variables than some of the other species distribution models, because it's accounting for the covariation in climate variables rather than treating them all separately. But yeah, that's a good point. There are probably some more practical variables to use. I would really love to have maps of temperature extremes, because that's where we could start taking this from a correlative model and then applying thermal limits to the actual model. So I think there's potential to get it sort of a hybrid between process-based and correlative. It still won't have growth indices like Climex has, but we could potentially apply those cutoffs if we have the data set. And if you know where there's a global data set of climate extremes in a raster format or some sort of spatial format, I would be very excited to talk to you.
No, this is really good. So I don't see any further questions on the Q&A or on the chat. And so I think that we can conclude this webinar. Frank, you okay that we conclude now the webinar? I think that it was an excellent presentation. I feel I learned a lot. So thank you so much for this. As I said before, this webinar will be available in 2 different YouTube channels, the International Pest Risk Research Group and also the International Seed Federation. And of course, if you have any questions, welcome to send to any of the sources, and we can always communicate with Joseph afterwards. And it was a truly pleasure to have you today, Joseph. Thank you so much for the presentation. Thank you for everybody who turned up today. Really appreciate. Thank you, Frank. I know you are suffering with this a little bit of a problem on your vocal cords. I am with you as well. But thank you so much for being here. Thank you so much, Joseph. Thank you to everybody. Looking forward to see you all the next time. Take care, everybody. Bye-bye.